So, okay. So, before I begin today, just a few announcements that I wanted to make, like I normally have every class. There's going to be your projects are going to be due next week on Friday, the report for the project. So make sure you start working on that if you have not already started working on that. Make sure you work with your group. You saw the grading for the abstract, and we paid a lot of attention on things like formatting and everything. So make sure that you follow all those guidelines and follow exactly what's given in the instructions for the project. And after you guys submit, in one of the lectures, I will just kind of briefly touch base upon what you guys should be doing for your presentation. So we haven't talked much about the presentation part, but we will do that once your reports are going to be submitted. As always, this week also you're going to have the learning activity. You're also going to have a homework and a quiz. So again, make sure you submit everything on time. I don't know why I still see that there's almost 6 or 7 people who never turn anything in, or they turn one of the assignments, not the other one. So don't do that. We are almost past the half of the semester now, so it's important that you start turning in all the assignments and everything, so that so that your grades don't get affected. Anybody has any questions or any concerns? Anybody online has any questions or any concerns? If everyone online want to turn off their mic and camera so we can make sure the bandwidth is appropriate. Anybody has any questions online? I know that I have been getting a lot of emails from groups that one of the group member is not working or not contributing. If that's the situation, you can always let me know, because I can assign grades individually to the group members. I don't have to assign the grades all at the same grade won't go to all the group members. So if you think that there's 1 or 2 group members not really contributing enough, then you can always let me know and I'll adjust the grades. If I don't hear from you, or if you don't put a comment, because there's a comment box when you submit your assignment, whether it's learning activity or whether it's project related stuff, just put a note that this person didn't contribute, and I will check with that person to make sure that they didn't contribute. And most of the times, either I hear from them saying that yes, they didn't, or I never hear from them. So that I just assume that they did not, and in that way, the people who worked the most would get most of the credit. So make sure you do that. So at least in that way, I am aware of any issues that are going on. Anybody has any questions? We also have a bunch of prospective students joining online today. I think some of them are already here. Today is actually, this week is actually engineering exploration. So if you want to,
No, I go to
mute your mic.
Doctor far.
Okay, I got muted too by mistake. So this week is the engineering exploration week. So there are prospective students who normally would be here on campus and visiting labs and visiting classrooms and things like that. But the situation that we are in, these students are actually joining by teams and kind of participating in this lecture. And I know some of the students who are here, I'm going to meet you guys later on this afternoon for the biomedical engineering session. Any questions before I begin today's lecture? Anybody has any concerns or anything that I should know? Okay, and I'm going to repeat this again. So, we have very few people in the class right now, which actually is great for a lot of social distancing, but there's less than what there should be. So if people don't want to come on campus anymore and are only going to attend the lecture online, let me know, send me a message, and I will move you to just online. So in that way we have accurate count of people who want to come in on campus and who don't want to, want to attend online. So just let me know if anyone who wants to move from on campus to all online. Okay, so I'm going to share my screen and start. So once I share my screen, just let me know if you guys can see my screen. Oops, that's the wrong way. Okay, just going to share my screen. Okay, everyone can see my screen, even online?
Yes.
Yeah.
Okay, great. So today we are going to continue talking a little bit about the electrical engineering concepts, and we're going to talk about biosensors. And we're going to spend some time talking about a couple of very commonly used biosensors as well. So today's lecture kind of ends, our introductory part of different, well, this course is an overview introductory course anyways. So from next week onwards, next week we have a lecture on, pressure and temperature measurements, especially for medical applications. And after that, we have all the guest lectures which are going to be there. So we have a guest lecture on intellectual property issues in biomedical engineering. We have a guest lecture on ethics. We have a guest lecture on technical presentation or STEM communication. How do you talk about science? How do you talk about engineering? So we'll have those 3 lectures. By the time it's only going to be fall break, so you guys will all go home, and then the rest of the semester, the 3 lectures that we have left, or maybe we have 2 lectures which are left after that, that will be all online, where we'll be working on the project part. And then you have the final exam. So today I want to talk a little bit about biosensors, and I'm sure that you guys have heard about biosensors, or in general, sensors are something that we hear quite a bit about, because sensors are the ones which we use to sense something, as the name suggests. A biosensor is specifically the one that actually is going to measure a biological activity. It's going to sense something that's biological, or it's going to sense something where a biological reaction is actually happening. So you have 2 different things that might react, and the sensor might show that this reaction has actually happened. And we'll talk about some of the terminologies which are commonly used for biosensors in a bit. But biosensors have been used significantly in, well, if you think about the applications of biosensors, there has been a lot in sort of the environmental area. There are sensors, even something like a smoke, a smoke detector. It's actually a sensor. It can actually detect the smoke. Carbon monoxide detectors, it can detect carbon monoxide. So those are sensors. They're not really biosensors because you don't really have a biological reaction that's going on. But a lot of environmental sensors are biological. There are sensors that you put it in a wastewater and it tells you if there is a bacteria or not. And it also sometimes tells you the concentration of the bacteria. So you know that if you need to treat this wastewater even more to remove those contaminants. From a biological applications, a lot of these biosensors are used for disease monitoring. They are used for drug discovery. They are used for, sort of looking at indicators in biological fluids such as blood and sweat and urine and saliva. The idea is that you take biological fluids, you put it on a sensor, and the sensor does something because there's a chemical reaction going on. And that chemical reaction may be as simple as producing a color change, and that's how you know that if a person has a disease or not. Or it could be some electrical signal that's actually formed. So you see like a graph or like a chart that shows up with peaks and valleys. And based on that, then you can kind of detect what is happening to that person. Is there a disease? Does that person have a disease or not? So that's a very simple sort of explanation of what biosensors are. But biosensors involve a lot of different components. And here is a cartoon which basically shows all these different components which are there for a biosensor system. So obviously, you are going to have an analyte, and let me go to the next slide, and I might go back and forth because this is where I have, it's a very crowded slide because I kind of explain about all of these different components which are there. So we have analyte, bioreceptor, transducer, electronics, and display, and we'll talk about those, and I'll go back and forth. So the most important part that you need to know is the analyte. Analyte is what you need detection. So for example, if you're trying to detect a certain kind of a protein that's produced because a person has a disease, then that is your analyte. And you need to know that analyte so that in that way now you can design your sensor that can actually detect the analyte, and not just detect, but also tell you the amount of that. And in that way you know how much that disease has actually progressed. So glucose sensors are the most common type of biosensors. And so glucose is actually an analyte because those sensors are actually detecting glucose, but they're also measuring the amount of glucose which is in the blood. So the idea with glucose sensor is that the analyte is actually going to be your glucose, or the one that you want to detect. Now, analyte is the one that you want to actually detect. But how do you detect that? You detect that with something called a bioreceptor. A bioreceptor is a molecule that specifically is going to recognize an analyte. So you have your glucose and you have some, and glucose can only interact with that something else, and that's how the reaction happens, that's how the sensing happens. So what it interacts with is basically the bioreceptor. And bioreceptors can be many things, so it could be small molecules, it could be cells, it could be DNA, it could be antibodies. And the way they interact, so the process in which they interact is almost like a lock and key kind of a mechanism, that only if you have glucose, it's going to interact with the sensor. If you have something else, it's not going to interact with the sensor, because if something else interacts with the sensor, then you end up getting a false result. So you have to have that very specific interaction between your analyte and your bioreceptor. But what happens when an analyte interacts with a bioreceptor? You have this lock and key kind of a mechanism. You will have some response. You would have things like change in color, or change in pH, or change in mass. And those are the things that you would basically measure to be able to know what that reaction is going on. And so this interaction is a very specific interaction, and many times we use the terminology called biorecognition. Biorecognition means recognizing, and it's only recognizing glucose. It's not recognizing anything else. And so you have this bioreceptor, and with analyte interacting with bioreceptor, you will have some things that are going to happen, the biorecognition process. So you have light, heat, pH change, mass change that happens, but how do you detect that? And how do you actually analyze that? So for that, you have a transducer. A transducer is an element that actually converts one form of energy to another form. So for example, with the chemical reaction, you have heat that's produced. This transducer is now going to convert this biorecognition event that has happened into something that you can measure, which means that it's going to show an electrical signal or something like that. And that electrical signal is something that you can capture, process, and get a number, and tells you that, oh, this is what the value of that analyte was in biological fluid like blood or saliva or something like that. So these transducers, they are mostly converting into optical signals. Mostly converting into optical and electrical signals, because those are very easy to process. We know a lot about how to capture these signals and how to process these signals, and we have a lot of instrumentation that has been developed to actually collect these optical and electrical signals. So once that transducer has basically converted the signal, and the signal, when it's converted, it's always converted with some proportionality. So you do have some sort of a relationship between the original biorecognition event and what the signal that's being transduced by the transducer. Then you need to have an electronics, because electronics are the ones which process that signal and show up as a visible signal, which means that you can see, like a peaks and valleys kind of a chart, or shows up a number. When it shows up a number, there is always going to be some sort of algorithm that's put into the system that converts that signal into a number. So there's different ways in which the signals can be processed, and then they can be, and electronics actually do that, but then you also need a display. So many times these biosensors, they have a tiny display, and then you can see a number, or you can see something that tells you, is this person is sick or not, or do they have more glucose, less glucose, if it's a glucose sensor. So that's typically what a sensor includes. Bioreceptor, well, analyte is important, bioreceptor, transducer, electronics, and then display. And the bioreceptor, is involved in the biorecognition process, transducer in signalification process, the electronics in quantification process, and the display just shows what that is. So it could be a number, it could be some colored chart, it could be some sort of a graph, And that's how you know that this process has happened, and you can also quantify the amount of the analyte that you're trying to get. So let's talk about different classifications of biosensors. So biomedical sensors, they're usually classified depending on what kind of reaction they are going to be involved in. They are typically physical sensors, electrical sensors, or chemical sensors, depending on what kind of application it's going to be. Biosensors, you can have further categories as well of biosensors, depending on what kind of bioreceptors you are using. So you can have an antibody sensor, you can have an enzyme sensor, you can have a cell sensor. So depending on what you're trying to do, you can further classify these biosensors. But in all of these sensors, you do need to have some sort of a transducer, which is going to now convert that reaction that's happening into some sort of a signal that's easy for us to detect and easy for us to convert into something that is a useful number that tells medical professionals that, oh, we need to start treatment or not treatment. Now, it's really important that there has to be some sort of a biological reaction for a biosensor. So for example, pH sensors, they are not really called biosensors as such, because in pH sensors, you don't have a biological sort of a reaction. You have to have some sort of a biological reaction to be able to call as a biosensor. pH sensor is simply working on a chemical reaction. It's basically a chemical sensing. It's not really sensing a biological process which is there. So sometimes, in classification, we would not consider pH sensor as a biosensor. For all practical purpose, people use the word biosensor very interchangeably, and it's absolutely fine to, do that. But the principle of how that sensing works, this analyte and this bioreceptor interaction that happens, that's the very important part, and that's what it decides if it's a biological sensor or not. But just wanted to give you guys, that, you might hear some people say blood sensor is blood pH sensor is a biosensor. It's really truly not, but it's okay because it's, sometimes we use these words very, very interchangeably. There are many different important parameters in sensors that we should be considering. So one of the important parameters is the sensitivity of the sensor. I'm going to go through a bunch of these different ones. So, and a bunch of these different ones have graphs and things like that. So I'll talk about that. So what does a sensitivity mean? Sensitivity is basically how sensitive your sensor is. But what does that mean, from a technical perspective? Well, sensitivity is defined as a ratio of the output change for a given input. So you're inputting something, what your output change, and then that tells you what the sensitivity is. So whatever you are inputting, if you divide that by whatever you are inputting and you take whatever output you are getting, if you divide that output with input, you get what the sensitivity is. So in a very simple way, if you have a graph of input versus output, sensitivity is just basically the slope. Now this is assuming that it's a straight line, right? If there is a direct proportionality between output and input, which may not be the case all the time. Like for example, you see that eventually the curve, it's curving here. So you always look at the sensitivity in the straight line part, and then you decide that it's sensitive in this region, and you know that there is a direct proportionality between output and input. High sensitivity means that small input causes large change. And that's what we typically want, right? If you're trying to detect something very, very small amount, but the signal that generated should be large enough so that you can actually measure it. If the signal generated is small enough, you have errors. And so that's why the sensitivity is going to be, very important. So a temperature sensor may have a sensitivity of, for example, 20 millivolt per degree C. What does that mean is that the output of the sensor will change by 20 millivolt per degree centigrade. Celsius temperature, which is actually pretty good, because by changing just 1 degree, if the value changes by 20, very easy to detect it. If by changing 1 degree, the value only changes by 0.01, you're not going to be able to detect anything. So that sensitivity is going to be very important. As I mentioned, that typically it's a slope, and you may have non-linear behavior, which means it's not a straight line. And when it's not a straight line, you really need to know exactly how that behavior is. And for different ranges, you define sensitivities in different ways. So for from this range to this range, this is what the sensitivity is, and from this range to this range, this is what the sensitivity is. Sensitivity can also be defined as the smallest change in the input quantity that will result into a detectable sensor output, which means the same, that what is that smallest change in the input that will result in a change that you can actually detect it. So at the end of the day, you want to detect what's basically happening, and that's what the sensitivity means. There are a few other parameters. Range. A range of a sensor is basically the minimum and the maximum operating limits. So many times when you use a sensor, it would say minimum it can detect this, maximum it can detect this, because that's a range in which you know it's most accurate, it's most sensitive. Accuracy is another term, as I just mentioned the word accuracy. Accuracy refers to the difference between the true value and the actual value. And you need to know that because you measure something with a sensor and it throws out a number 100. But is it really an actual value? Is it really a true value or is it an actual value? The true value may be something different, and the accuracy basically tells you how different what you're getting is from the true value. And you need to know that as well. Typically, accuracy is expressed as the ratio between the difference of the true value with the value that you're actually getting in the full scale. So you have to kind of see the accuracy for not just one particular range, but for the full scale of how the sensor is operating. The other important parameter is precision. Precision is the degree of reproducibility, which means you're using the sensor again and again and again, and is it giving you the same results? Reproducible results are important, because if you don't get reproducible results, obviously there's a plus minus, some error is always going to be there, but within that error of margin, if you get reproducible results, then you know that your sensor is working fine. And precision is not accuracy. Precision is the reproducibility. You may get the wrong value, but are you getting the wrong value again and again and again? That means it's very precision. But accuracy means how different the value is from the true value that it should be. And so measurements can be precise, but they may be inaccurate, you know. So you could have precise measurements, but maybe the values are not going to be that accurate. Then we have resolution. Resolution, I'm sure you guys have heard quite a bit, you know, especially we talk resolution, you know, when you look at screens and things like that, we talk about resolution. But from a sensor perspective, what does resolution actually mean? So when an input quantity is increased, the output may not change until you have a certain input that you are giving. And so resolution is defined as the smallest distinguishable input change that can be detected with certainty. And so that would tell you things like, well, the resolution is, you know, 0.5. So as far as you have 0.5, you can detect it. Less than 0.5, you may not be able to detect. 0.5 is the smallest, and I'm just giving a random number, 0.5. It could be, you know, concentration of glucose or, you know, whatever that you're looking at. So resolution means what's the smallest you can actually detect with an input that you can actually give. Resolution is important because many times you want to accurately measure. If a sensor gives you number 100, 101, 102, that may not have a good resolution, but we would need sensors that can measure 100.1, 100.2, 100.3. So that 0.1, 0.2, 0.3, that difference is 0.1 between them. That's the detection that you can resolution of the sensor which is there. Reproducibility is important. Remember, we talked briefly about reproducibility, but reproducibility is how close the measurements are when they're repeated over time. When the range of measurement is small, the reproducibility is high. So for example, a temperature sensor may have a reproducibility of plus minus 0.1 between a certain range, but more than that range or less than that range, you don't have that kind of a reproducibility. And reproducibility many times also depends on the range. So what that range you're trying to actually measure. And then we have offset. Offset is the output value when the input is 0. I'm going to go back to this graph here so I can actually show you what that offset is. So you're not really doing anything. You're not really putting any input, but the sensor has an output. And you need to know that, which is basically the offset. That means that at 0 input, you still have some output, which is inherent to the system. That's how the sensor behaves. And now when you start giving input, you're going to see that the output is going to change, but you have this offset which is there, and we need to know that offset. These things are important, especially when you're going to measure, when you're going to develop algorithms that go into the electronics to convert these into numerical values. So you need to know what that relationship between output and input is going to be. We already talked very briefly about the linearity, which is that straight line region. I'm going to go back to the slide so that I can show you again. So this is the linear region which is there. So the linearity is a measure of the maximum deviation of any reading from a straight line calibration. So we always try to do was calibration, where we know that we give a certain input and we get a certain output. So in that way, we can create a calibration curve. In that way, now if a sensor throws out some input, we put something in the sensor, then we can go back and see what that output is. Or if the sensor is outputting something, we can go back and see what the input should be for getting that output. So that calibration line is an important aspect. And as you can see that it could be a straight line, but eventually it might actually curve off. And so that's a very typical behavior with a lot of different types of sensors where you do not have this straight line relationship. So many times we would refer linearity as a percentage, and we would say that this sensor is 90%, has 90% linearity, which means that for 90% it's linear, after that it's not going to be linear. It's important to know that linearity is not so much of an issue these days because we have better computer programs to actually model that behavior. And so we can fit in the algorithm into the electronics, so we know that more than this concentration, it's not linear, so it can read the values much more accurately. We would like that there is linear behavior, but many times if you try to assume linear behavior for certain kinds of sensors, you don't get accurate measurements. So we have to take into account that sensor behavior is not going to be linear, and the input-output relationship is not just a straight line. It's going to be maybe partly straight line, maybe a curve, maybe an exponential curve, maybe a polynomial kind of a curve. And we need to know that so that we can accurately find out what the values are going to be. The other important part is the response time, because obviously response time is going to be important. You use a sensor, and if the sensor takes 2 hours to get the result, that may not be of advantage. So you want that response time to be quick enough so that you can get the value. And so response time indicates how much time it takes for a sensor to accurately reach the steady state and give the number that you need. So for example, it may take 20 seconds for a temperature sensor to reach 95% of its maximum value when you change the temperature by 1 degree. What that means is that you change the temperature by 1 degree, and within 20 seconds, the value that you get is 95%. And that response time is important because in that way you can have fast detections. You don't have to wait for a really long time. Imagine if a glucose sensor would take 2 hours when somebody uses it. For those 2 hours, they can't do anything. They can't eat. They can't decide, what kind of medication or insulin they need to take. So you want very quick response times, and quick response time indicates that the sensors are responding quickly to whatever changes they are exposing to. And so that's a very important parameter as well. There's few other things that we are going to talk about sensor specifications. There's something called drift. Drift refers to the change in the sensor reading when the input remains constant. So you apply some input, you get a value, but if you wait, that value keeps on changing. And so drift is something that happens, and how do you quantify or how do you take into account for drift is you basically run many calibration, and you determine what exactly the behavior is. In that way, drifting from this value to that value, and you know that this is what the range in which you are going to get the values. Input-output relationship over time is going to change. So if you use sensor for like 24 hours, and this is typically true with a lot of environmental sensors because they put it in and they just leave it in and the data is being collected. Over time, the values might start drifting and you may not get accurate value. So you need to know what that drift is to be able to actually predict what the accurate values are going to be. So let me go and kind of talking about this drift. So there are kind of ways in which you can have drift. So one is this is what the ideal is, but over time you could have values that would range in this way. This is really you know, related to the sensitivity part, because your sensitivity is changing plus and minus, and depending on your input, your sensitivity drops. Basically, your value could be anywhere in that region. It can happen, sensors can work this way, or you could also have offset errors, which means that, you know, ideally we would want to have input 0, output 0, which is typically not the case. We always have input 0, but output might be something. So your curve may look like this or may look like this, and you still have a range which is there. So ideally we would like 0 drift. That doesn't really happen. You're always going to have drifts in sensitivity and drifts in the offset. That's going to happen. But knowing these values, you can feed that into your electronics so that the final value that you get takes into account all these errors which are there. A couple of other things which are important. One of them is hysteresis. Is basically how the input and output relationship is. And here is a very simple, you know, sort of example. You have a sensor with input and output. As the input increases, you see that the output will increase. It will reach value. And after this, now if you remove the input, it comes back this way. So it doesn't follow the same path that it actually took. It's taking a different path coming back to the rest state. And this is very common. We will need sensors that would do like this, because if you have to use sensors repeatably, that means we will remove the input, the value should come back to 0. And we expose again, the value should go back and it should come back. So that's sort of the repeatability aspect of the sensor. And so this kind of behavior is very common. It's very direction dependent. It's very much dependent on sort of the input, how you are giving the input. And you can see here there is an offset on output side, and here now we have an offset on input side, and that's happening because the return path is not exactly the same as the path that it's actually taking. So these kind of hysteresis very commonly happens in a lot of biochemical reactions, and so we have to take this into account as well. Why is this going to be important? Well, when you bring this back to the rest, and you have to use a sensor again, now it's going to start from here. It's not going to start from here. It's going to start from here. It's going to go up, and then it's going to come back here. Or it might go back somewhere here. So you need to know that every time you keep on using that sensor, how much it's varying, and you have to take that into account when you are actually finding the final output and what that final number for the sensor is. So I think I gave a lot of information on sensor. Anybody has any questions? Anybody online if has any questions about sort of the basics? So I gave everything that is possible to know in a very simple way about sensor. These are all the different parameters which are important for sensors. Any questions? Oh, there is a question in the chat. So let's see. Do you want to just... The hysteresis would set the output to 0 or output to a certain value below which it cannot go below. So it can kind of reset it, but the thing is repeatably that you use, you might even see your offset is changing, you know. And once that offset changes significantly, you know, the sensor is of no use anymore. So you are not going to use that sensor anymore. So yeah, but most likely it's not going to, because then you are going to end up, you know, eventually depleting that sensor properties, and at some point the sensor is of no use. Many sensors you see in the market are use and throw sensors kind of thing, so one-time use. And the reason why they're one-time, because you cannot reproducibly produce the same signal that you had the first time, because either it's because of hysteresis or accuracy or the reproducibility. So all these things, you know, depend on, you know, what kind of detection you are doing and what kind of sensor you have. And many times people intentionally develop these one-time sensors because they're cheap. You don't have to worry too much about the repeatability or reproducibility. Just use it one time and you get rid of it. So it depends all on what kind of application you want to use, but continuous sensors, especially for environmental applications, this is going to be a very important aspect. Okay. Is a pregnancy test a biosensor? That's one of the questions. It is actually a biosensor because it is trying to detect certain kinds of biomolecules by showing a color or showing the change which is there. So certainly, yeah, it is a biosensor. Yes. Okay. So what are some of the applications of biosensor? Well, they are used in many different areas, and I think I already mentioned enough about these areas. But just to give you again, they're used for drug discovery, disease detection, environmental monitoring, soil monitoring, food monitoring, toxins, water quality. There's many different applications where biosensors are being used. One of the recent advances that has happened in the field of biosensors is because we have now ability to make nanomaterials. So nanotechnology has helped significantly the field of biosensors. And the reason why nanotechnology has been so much influential in biosensors because now you can reduce the size of sensors. So when I mean reduce the size of sensors, it's actually the sensing element. It's not the whole sensor itself, but what is that element or what's a part of that sensor that's sensing. That has helped to reduce the size. And when you reduce the size, you have a much better noise signal to noise ratio, because now your surface area that you're exposing to your analyte is much smaller and much controlled than what you would have. Also, you need much amount of samples, which is really good because you don't want to draw like 10 mLs of blood if you want to do something. If you can just prick the blood and get just a drop of blood or, like 10 microliters of blood, that would be perfect because in that way you're not really doing invasive or it's not an invasive process to get the sample. Cost of assays is also less because now we are using less reagents. So whenever you're doing sensing with these nanosensors, you need very less amount of reagents. And by moving to that nanoscale dimension, the biggest advantage is that your surface to volume ratio has increased significantly. Your surface has become dominant. Volume is not that huge a big deal anymore. It's a surface which has become very active. You can also now prevent non-specific attachment. That means you want the analyte to attach, but attach. If anything else attaches on the sensor, you're going to give a false positive result. And you don't want that to happen. And so there's a lot of research which goes on in these nanomaterials, which are used for sensing applications, and how we can make the surfaces of these sensors so that they're very specific to what kind of analyte you want to detect. One of the commonly used is graphene. Graphene is carbon, basically. Everyone knows what graphene and diamond is, right? Hopefully you guys know, diamond and graphene, they're all carbon. It's just that how the atoms are, how the atoms are arranged. You have diamond, the hardest material, one of the hardest naturally occurring material, and you have graphene, which is a very soft material, which is sheet-like structure. So this graphene can be oxidized from graphene oxide, and people have used graphene oxide for biosensor applications. Graphene oxides, carbon nanotubes, they're all carbon-based material. And these materials, they have much lower detection limit, which means that because they're nanoscale, you now detect very, very small changes in the analytes that you're trying to detect. So having talked about all of these things, what are the challenges in biosensors? Well, obviously identification of an analyte. You need to actually identify an analyte that is important, and there has to be advantages of a biosensor over traditional methods. So traditional method would be you collect a sample, you go to the lab, and you do a bunch of reactions, and you do a bunch of calculations, and you end up with some values that tells you, yes, there was this contaminant in wastewater, yes, there was not a contaminant. A biosensor would be something that you take, just stick it into it, it's connected to some sort of a screen. A few seconds, a number shows up. If the number is above the threshold, you know that there's contaminant. If it's below, you're fine. So you have to kind of think of what would be a better option. But the other practical aspects about biosensors is the storage aspect. You make a sensor, how long can you store it? If you can store it for a really long time without the sensor degrading by itself or decaying by itself, that would be a good thing. So the stability is important, the cost is important. Cost gets very complicated, especially and very expensive, especially when you use nanomaterials. So you would think that you're using nanomaterials, small amount of material, it should become cheaper. But the fact is that it gets more expensive if you have materials, because making those materials is much more complicated. At that point, it's not the amount of material which is the cost driving, it's actually going to be the fabrication process that you actually use. And obviously, you have to think about hazards and ethics which are related to that. So let's talk about glucose sensor, which actually is one of the most commonly used sensors. Glucose sensor could be part of two different types of systems. It could be a continuous glucose monitoring, where the glucose is being monitored continuously. Typically, those are inserted under the skin, and it allows to sort of measure the glucose level real time. So you can actually get real-time glucose measurements. Now the sensors are very well developed. So if you have something sensors under the skin, it can wirelessly transmit to a device. It could transmit to a phone or a watch or some sort of a wearable system. And you just have to press a button and you can just see what the glucose levels are. So these are actually very expensive systems. These are not very simple systems. They are invasive systems, which means you do need to put it under the skin. So it actually goes under the skin, involves some minor incision and things like that. And here is a cartoon which kind of shows. So you put this and you have this blood vessels which are there. And you basically would have glucose that would detect. Many of these are not really blood vessel related, but they're also sweat related. So the sweat can also measure the amount of glucose. It may not be the exact amount that is present in the blood, but it can still measure it. But in an indirect way, you can tell if more in the sweat, the amount of glucose more is in the blood. So there's many ways in which you can actually do that. But this is a real-time thing. And for a lot of people who have fluctuating diabetes, this makes sense because they need to know how that is going up and down. So again, for a sensor, again, I'm just going to briefly touch base on what we had talked before. So for a glucose sensor, again, you have the same components. You have analyte. Analyte is going to be the substance which you are trying to detect. In this case, it's actually going to be glucose. It's not just glucose, right? When you're in the blood, you have many other things, but the sensor has to only detect what that glucose is. Then you have a bioreceptor. Bioreceptor is what, again, as I mentioned, interacts with the analyte. And the bioreceptor in this case is actually an enzyme. And these enzymes, enzymes are proteins as well. These enzymes, does some reaction with glucose. And so when you have a blood drop which has glucose comes in contact with enzyme, there is a reaction that happens up. And then you have this transducer which converts that energy into the form that we can actually measure it. So just to give you this cartoon, so you have this glucose. This glucose interacts with the bioreceptor, almost like a lock and key kind of mechanism. When that happens, the transducer produces a signal and the display will show you what the value is. And that's typically how the sensor would actually work. A lot of earlier generation glucose detection systems were just change in color. So you would see there is a change in color that happens, and then you have to have a process to convert that color into a measurable value. But nowadays, the systems are so well developed that it just throws out a number on the display, and you know that, oh, well, this is high, this is low, and if it's high, it beeps or does something, Then that way you know that the person's glucose is more than what it should be, or it's less than what it should be. So the principle is same exactly as what we talked before, except now the analyte is glucose, the bioreceptors are certain enzymes. You have a transducer, which basically converts whatever reaction is happening into an electrical signal, and then you have a display which is going to now show that value. Depending on what you have programmed in the electronics, it shows up a number. But many of these also show up a graph. So you see the graph going up and down, and you click a button, it shows you the value as well. So it can show you up and down graph also, or it can show you the value also. There are different types of glucose sensors. Glucose sensors, as I mentioned, they come in a continuous form, but the more cheaper ones are these discrete glucose sensors, or these test strips, where you can just prick and put a drop of blood. Both use very similar process. The process is very similar for both. If it's a wearable sensor versus a strip sensor, it's still going to be the same sort of biochemical reaction that's actually going to go on. And they use an enzyme, and this enzyme actually reacts. In the next slide, we'll see what these enzymes are. Typically, what you do with these strip kind of sensors is that the enzyme is coated on the strip. So when you put a drop of blood, the drop is reacting with the enzyme which is coated. And then in that way, it might show a color, or you may have to use another system to put that strip in, and it quickly tells you, what the amount of blood glucose is. So different types of enzymes that have been used are glucose oxidase, glucose dehydrogenase, nicotinamide, adenine, dinucleotide, bunch of different ones. Once that reaction takes place, you basically would have that energy conversion that would happen through a transducer, and then you have a glucose reading that's actually going to be provided. So these blood glucose sensors have been used for a long period of time. Especially these coated enzyme strips, they're very cheap. I mean, cheap is also a word, but much cheaper than a continuous glucose monitoring system. But these are also single use. So as I mentioned earlier, some of the times you look at the cost and see if it's cheaper to actually produce something that is one-time use. Continuous glucose monitoring would be way more expensive, whereas these strips are really cheap. The big difference is continuous monitoring would continuously monitor the blood glucose, whereas the strips is whenever you try to do it, And so if people's blood glucose fluctuates a lot, they may end up using many strips, and it might be better for them to have a continuous glucose monitoring. Many times, you also have sort of electronics built into these kind of chips as well. So these electronics can also communicate with the glucose meter. There are new developments that have happened is similar to this continuous monitoring, which are under the skin, but you have these strips which have some electronics built in. You put a drop of blood. You don't need a machine to put it in. It wirelessly transmits to a reader, and you'll know, what that blood glucose value is. And over the time, these blood glucose meters have become much more affordable. They used to be very expensive, but they're much more affordable. They provide sort of on-demand values, not so much of continuous monitoring, but it's also almost like an on-demand. When you feel like you want to measure it, you do. And many times people measure before eating, so they know that how much is the blood glucose level. They would do it after, so then they know that if the insulin that they took is working or not. So those are, and again, this is something that doesn't have to be there on the person all the time. So the continuous glucose monitoring has to be implanted, whereas this doesn't need to be implanted. It's just once in a time. So in contrast, the continuous glucose monitoring, again, it's a very similar process. You do have a sensing element which has its enzymes on it. It's a wearable sensor. It measures glucose levels 24 hours a day. And many times people get these kind of continuous monitoring devices when they're actually admitted in the hospital. So especially when they're admitted in the hospital and they have diabetes and they're going through some surgery or some treatment, this is what's done temporarily so that they can monitor the glucose as they're going through that process. Many people also decide to have this, at home, so that at home also they can actually detect it. And it can detect for several weeks. Now, at some point, the sensor will not work, so you'll have to go back and remove it and put a new one, and that could be a little bit invasive procedure if you have to do that every few months. But there are new sensors which can be worn for months. There are sensors which say, 5 months, 6 months, very easily you can actually wear it. So people, when they just decide whether it's convenient for them to prick and get the blood whenever they need to, or they would like something which can continuously actually monitor. The best advantage of this continuous glucose sensors is that they wirelessly transmit, and many times the medical professionals can also kind of monitor your glucose levels, and can tell you that, hey, this is happening. Don't you need to take more sugar, or your insulin is not working, or something like that. So for a lot of people who don't understand some of these systems, this remote monitoring can be of significant advantage. So here is how these continuous glucose sensors work. Typically with a needle, you can insert this under the skin. There are needle-free options as well. So this is an example of a needle-free option. You see something like this that's outside the body, but there's a very small part that actually goes inside the body. This is from a company called Waveform, which makes these sensors, and they put it typically in the abdomen or the back of the arm, but the manufacturer tells you where it's going to be most accurate. The only thing that people who have these sensors should be careful is not to move it around, because if you move it around, it can cause infection and things like that, and the accuracy might go down. So sometimes they put like band-aids and things around so that it doesn't move around. So it could be inconvenient for some people, and that's why many people just choose those strips because they're very easy, and they don't mind, getting a drop of blood or a few microliters of blood every time they want to test it. But people who have very fluctuating diabetes, they would probably want to use something like this. So that's all I have for today. Anybody has any questions? Any comments? I have not looked at the chat, but was there anything in chat that I missed? Okay. Okay. Anybody has any questions? So remember, quiz, homework, learning activity. Everything is posted as usual. It's all going to be due. Quiz on Friday, learning activity, and the homework is going to be due on Sunday. So make sure you look at that and submit on time. Okay, if you guys don't have any questions, I will see you guys next week.